Hello, my name is Thira Faruangsaeng. I'm a master degree student from Regenerative Dentistry and Special Patient Care, Chulalongkorn University. Today, I would like to present my research project titled Gene Expression in Human Dental Pulse Cells of Mandibular Second Premolar Teeth. My research advisor is Associate Professor Dr. Thira. Tooth development is controlled by complex reciprocal interaction between genes and signaling, and share similar pathway with other ectodermal organs. Calcified dental tissue comprise enamel, dentin, and cementum. The main tooth component is dentin, produced by odontoblasts. Many studies have demonstrated that odontoblast differentiation involves several signaling such as bone morphogenetic protein, fibroblast growth factors, and wingless signaling, and many transcription factors such as RANX2, MSX1, MSX2, and PAX9. One of the most common congenital missing tooth is the mandibular second premolar. Regenerative dentistry is a trending concept that uses dental stem cells to develop new tooth and restore destructive tooth structure. Cell-based dental tissue regeneration requires cells from adult teeth together with an understanding of molecular mechanism and biological process involved in tooth maintenance, function, and vitality. To date, there is a very limited knowledge about gene expression in mature adult teeth. And the objective of this project. This study therefore aims to investigate molecular characteristics of adult human dental pulse cells, especially significant genes that are involved in odontogenesis. Definitely, we need to increase genetic knowledge of mature adult tooth, but how? Nowadays, there are many methods to study gene expression, but the two popular methods are quantitative real-time polymerase chain reaction, microarray, and RNA sequencing techniques. qRT-PCR is commonly used to analyze the expression of a few genes with a known sequence, and microarray used to measure the expression level of a large number of genes. The downside of microarray is the low dynamic range of the assay and the need for probe design sequence. And the third is RNA sequencing technique. It enables you to look at differential expression at a much broader dynamic range to examine DNA variation and even discover new genes or variation using just one data set. RNA-seq is considerably expensive. However, the data achieved from RNA-seq is tremendous and very beneficial for experiment. The second part is material and method. We obtain the dental pulse cell from mature permanent second premolar of 2 healthy participants and perform a transcriptome analysis to analyze gene expression profile. The research protocol was submitted for approval to the Human Ethics Committee, Faculty of Dentistry, Chulalongkorn University. Informed consent were obtained from participants. And the next is method. Tooth scheduling for extraction according to dental treatment plan were collected for cell isolation. The mandibular second premolar teeth obtained from 2 unrelated participants were employed. The data of teeth, tooth number, cast roots were photographed and recorded. Briefly, dental pulp tissue were gently removed and expanded. RNA were isolated from cell at passage 4, synthesized to complementary DNA, and subject for RNA sequencing. RNA-seq analysis was carried out using RNA-seq alignment and RNA-seq differential expression program. Reactome program was used for pathway analysis. And the next part is result. The RNA sequencing demonstrates that 17,968 genes out of 27,914 genes in total were expressed in adult premolar teeth. Here, the table shows top 20 expression genes in mandibular second premolar. The FN1, CO1A2, ACTB, CO1A1, and EEF1A1 were the top expression genes. The first one is FN1 or fibronectin. The fibronectin protein is involving in the organization of extracellular matrix and cell movement via actin organization in cell cytoskeleton. The second one is CO1A2, which encodes for alpha-2 type 1 collagen and play a role in collagen formation. This picture shows significant pathway according to the top 20 expressed genes created by reactome program. The related pathways are highlighted in yellow color. From top 20 gene expression, there are 8 major pathways involved. First, extracellular matrix organization. Second, immune system. Third, signal transduction. Fourth, developmental biology. 5, metabolism of protein. 6, hemostasis. 7, cell-cell communication. And 8, vesicle-mediated transport. And extracellular matrix pathway was the most involving pathway. The bar chart shows expression of genes related to odontogenesis with the level of expression on y-axis and the selected gene on x-axis. From 33 odontogenesis genes, 28 genes were expressed, while 5 genes were not expressed in a major adult premolar teeth. The final part is discussion and conclusion. Here we show that the top 20 expressed genes in adult human teeth consisting of FN1, CO1A1, CO1A2, TGFBI, IGFBP5, and TIMP3. These genes are important in odontoblasts and dental pulp cell activity, cell proliferation, inflammation, and organization of extracellular matrix. Interestingly, we demonstrate that genes related to tooth development comprising the BMP2, BMP4, MSX1, MSX2 are still expressed in adult human teeth, while FGF3, FGFH, LHX7, ALX3, and FOXI3 were not observed. This finding promote further study to understand gene expression and signaling that control cellular physiology of adult teeth and how dental pulp cells respond to external stimuli. This would advance our knowledge about genetic engineering and stem cell-based therapy in regeneration medicine. Thank you.
Thank you for your presentations. And questions are welcome from the audiences. Okay, while waiting, so I can give you some questions first. So from the data of your research, it's apparently like the genes that you extract is only from 2 samples. Would you please explain the reason behind this? And this result can is it variable enough to represent all the population of second premolars?
Because it is the experiment that for to find the gene expression profile that no one has done before. In addition, the researcher has a limited time to study and cell culture and the process for an ASIC took a very long time. This experiment is like a pilot study. But in the future, we will increase the number and the type of teeth for represent the 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 second premolar teeth.
So how many samples you plan to do this RNA expressions? Which is any previous study suggested?
No, but I will do this after before I start the next experiment.
Okay. Of the objective of the study, you say you just say that you're interested in the second premolars because it's the first congenital missings, right?
Then one of the most congenital missing is including third molar. Third molar is the first and the second premolar is the second. But there are many research that have report it's not the same. It's not the same. Maybe they report second premolar is the most missing, but some research said that the third molar is the most missing. And some research has said that lateral incisor is the most missing.
Okay. Some time left and no more questions from the audience. So I will have one questions more. So from the table 3, I can see that there were dramatically variations of gene expression related with odontogenesis between 2 samples. Do you have any explanations for this?
Do you mean this?
Yes, you can see the expressions number. Sometimes it's too manifold different from two twos. And and you use the mean count to plot the graph. So I want to know, is it normal to have so much variation between these amplifications? I mean, the number of genes. Because, for example, like FTH1 is like 48,000, but this one is like 350,000. Like this, it's so much different.
It is because I have a less sample, and
So is it possible that the criteria of inclusions of donated teeth do specifies any characters of the donated teeth to be similar or not?
Our teeth is obtained from the healthy participant and female patient and age around 20. But the difference between sample 1 and sample 2 is much, but I think it can decrease if I have more sample.
Okay. So from the sample selected, did you see the morphologies of the tooth before extracted or the size of the pouch chambers before you extracted the pulp?
Yes. Did you mean
that it's quite similar?
Yes, quite similar because it from the second premolar.
I mean, same
the root.
Okay. So 3 minutes left. So I can see that you have the genes, some of genes that didn't express, right? And you say it isn't expressed in adult second premolars. So I want to know if these genes appear in others.
Do you mean that again, please?
Can we suggest, you say that some of the genes disappear, right? In another. Yes. And I'm just asking, is it specific, is only specifying for these tooth or it's, which teeth have this gene? I'm asking. You say the second premolar, these genes is not shown. So I'm asking if this
it disappear in
other teeth or not.
the two sample. But if we have more sample, I think it may be more accurate than this.
Okay. So how much per sample? How much money you have to spend per sample?
How much?
You have, because you have to send this to the lab, right?
Around to 20,000 baht per teeth.
Sample?
Per sample.
That's a lot. So if you plan to do at least 10, you have to have like a lot of money for your research project. Is that right? And besides how it's difficult to culture or extract the pouch tissue, is it difficult?
Yes, it's difficult because
you say it take a long time.
Some sample I have a contamination and we have to search for the new tooth. And it's a COVID situation and the dental treatment is limited when the COVID is terrible.
COVID make our life difficult.
And it make it hard to do the experiment. And it takes around 6 months after I send it to produce the unacid data, and after 6 months I receive the raw data and analyze this.
Thank you so much for your informations. And I hope you can find grant for more tools for your next research. Okay.